All right, so we're going to get going with the Medicine Informing Novel Discoveries Mind series, and this is our second one for the astrobiotechnology. We did the Global Astrobiotechnology Hub event, but the astrobiotechnology theme we're going to continue on, really focusing on patient advocacy, being a community leader, but also seeing where science will take us in this unique environment in space. So I'm thrilled to be able to have our rock star, Sandra Dillon, here. So we're going to ask you to just come up to the front, and then we'll have you there just because. You see, you have to stand up for science. So Sandra Dillon and I have known each other for quite a long time. She was literally the poster child for CIRM, the California Institute for Regenerative Medicine, for Prop 14, just led to $5.5 billion, I'm just saying. So she thinks big, makes us all think big. So for those of you in the room who don't think aspirationally, talk to Sandra, because she will change how you think about things. Dr. Khalessi, you're on the panel too, just before you get too comfy. So Dr. Khalessi has to do things quickly because he looks after people with brain tumors and is very interested in neurorestoration, is our co-director of our brain tumor and neurorestoration program, and also was just the head of the Congress for Neurological Surgeons in the US. So really the top neurosurgeon, no pressure, Alex, because we know you don't feel pressure, he's a neurosurgeon. We have Jana Stoudemire. She's coming back to explain why space. We keep asking this question. What have we got from this rather expensive place? Is it paying off? And we've got our friend and community leader, Rebecca Moores, and her grandson, Keeve, who's going to get our friend Becky up here. You may recognize her. Her name is on the Moores Cancer Center, Rebecca Moores. Thank you so much. Thank you. Yes. Oh, wherever you like. Yeah. Yeah, it's going to be up to. And then we've also got Alysson Muotri while Becky gets settled here. Alysson Muotri is the director of the Integrated Space Stem Cell Orbital Research Center, which stands for I-SCORE, as in the hockey term, because you have to have a lot of shots on goal. Thanks, Becky. So we're going to start with a presentation from Alysson Muotri about advances in this unusual environment in microgravity. He actually instigated this all. We went, to see a launch that Alysson Muotri was going to have, and Jana was there, and she said, "Of course it'll work." And then we said, "Really though?" Because there were a few hiccups. Danny took us there. David Brenner was there. Tatiana was there. Sheldon was there. We said, "Is this really going to work?" Of course it did, because Alysson made sure that we really witnessed something spectacular. It didn't work the first time because it didn't launch the first time. It scrubbed at what was it, T-minus 30 seconds? And who didn't give up? Alysson did not give up because it's not in his lexicon. It's not in our lexicon. We don't give up. It's not a thing. That's a luxury that our patients can't afford. And because we're all going to be patients, none of us can afford that. We have to make new therapies and new diagnostics to go with them. So I just wanted to see how many people in the room are scientists? Wow, okay. How many people in the room are patient advocates? Okay, that's actually quite a lot. How many people work in administration? Well, that's quite a lot too, hey. Okay, how many people are in business? Okay, this is a lot. This is a cool group. We're going to get things done today. So speaking of getting excited about things, you're going to see some pretty astonishing science from Alysson Muotri. And without further ado, Dr. Muotri.
Thank you. Thanks, Kat. Thanks, Kat, for introduction. It's always nice to team up. I think always when we team up, there is good things that emerge. All right. So yeah, I'll be talking about growing human brains in space. There is a reason for that. But first of all, you need to know that space is a really harsh environment. It's harsh to your body. It's actually pretty bad and affects different systems in the body. And some of those, it's reversible. So after the astronaut comes back, kind of reverted back, and I think Imed mentioned about the muscle loss. But there is something that is really problematic, and that affects NASA a lot in all the idea of interplanetary colonization, which is the impact on human cognition. And this was first, I would say, perhaps better explained by one of the publications from NASA, is the NASA Twins project, where one of the genetically identical twins stay in space for about a year. The other one was the ground control here on Earth. And what you are seeing here, it is graphics to show accuracy on the cognition side, speed, memory, learning. And you have like 3 stages here: pre-flight, post-flight, pre-flight, during flight, and then post-flight. And the green one is the ground control. The blue one is the astronaut when he comes back. And I hope you can appreciate that even after 6 months when he's back on Earth, the cognition levels is still pretty bad compared to normal population and even compared to his brother. So why microgravity, why the space environment is bad to the brain? So this is a hard question because it's hard to study. How would you study the brain? We only have no invasive technology to do that. So we postulate something else. What if we send brain organoids? And these are tools that we use in the lab to understand human neurodevelopment. These are stem cells that can create a 2-dimensional model of the human brain. And if you've never seen it, so this is like what we do in the lab. So we can actually take stem cells, reprogram from people, and actually create these brain organoids. They grow as small as spheres, but they can actually expand to 0.5 centimeter in diameter. They contain most of the cellular types in the human brain. And you're going to hear a lot in the media about mini brains. And I really don't like this term because it gives you the false impression that we have a miniaturized human brain there. It's not it. But it is a tissue that contains, again, all the cell types and behaves and functions like the human brain. But there is a lot of things on the technology side that we still have to develop. So what Kat alluded was our first payload back in 2019. It took almost like 10 years to actually create that first payload. But then we launched it, and then we have like several other launches throughout the years. So I think we are in our 8th payload now. And basically, it's all in an autonomous box. We work with our implementational partners at Space Tango, where we have this autonomous box that can keep these organoids alive during the mission in space, requiring like almost no interaction with an astronaut. It's all fully autonomous. And the design of the experiment is the following. We grow the organoids here. We ship them to the Khalessi. So that's the Kennedy Space Center where we actually prepare the payload, and then we send them to the space station. After about 30 days, we collect the payload. It splashes down, and we open the payload. We get the stem cells or the organoids from there, and we do several analyses at the molecular and cellular level. We are now doing network analysis as well. And the biggest surprise is we see something that was alluded before. We see an accelerated aging. But this acceleration of aging is not linear. It's not exactly what happens here on Earth. It's actually a different thing. Because it's a different thing, it's neither maturation, it's neither aging, we coin a different name. We call it space-induced neurosenescence, or SINS. It's a new phenomenon. Nobody has seen it before. And because it's so important, we decided to study it at different levels. And as I mentioned, we can look at the molecular level. We can see what are the pathways that's being affected by space. We can even compare with other data sets from the gene lab and see what kind of genes are driving this senescence effect. But I'll give you like one practical example is when we start applying this to study neurological conditions. That was the idea. If I can speed up in a compressed time human neurodevelopment, perhaps I can understand better how these neurological conditions emerge. And if I understand them, I can block that. I can prepare some kind of a treatment. And the example that we have right now is with Rett syndrome. It's a severe neurological condition. These kids are born quite normal for the first year, and then after 18 months, there is a regression phase. And in this regression phase, they start losing all their abilities to talk, to walk, to move, and they end up in a wheelchair for the rest of their lives. We have better care now, so they're living a little bit longer. But in the past, the short life was during the teenager years. And when we send brain organoids from Rett syndrome, there's something that we observe is this activation of endogenous retroviruses. So these are viruses that are encoded in your DNA, and we all have them, but they are all silent now unless you age. Once you age, these endogenous retroviruses start to be expressed, And the cells, the immune cells in the brain, realize that they're being attacked by a virus, and they trigger an inflammatory pathway. And that's what we see here. This is like a cytokine called IL-6. It's a pro-inflammatory cytokine, high levels in the brains of Rett syndrome. So once we learned that, we thought, well, I mean, if these endogenous retrovirus are promoting the inflammation, what if we repurpose anti-retroviral drugs that already exist for Rett syndrome? Nobody thought about that before. And so that's exactly what we did. We took advantage of a mouse model for Rett syndrome. These animals are really sick. They die in about 4 weeks. And what you have here is the survival curve. And we start giving the treatment with an anti-HIV drug 1 day before they die. And we can expand the survival curve, which will be the equivalent of 10 years in humans. Not only we can extend the survival, but we can improve the quality of life of these animals. So they start recuperating all the weight loss, they start moving around, they start eating better, they become healthy again. So this is like a big surprise because no other no other medicine for Rett syndrome can do what we see here. So I'm glad that with Sheldon here, we are moving to a clinical trial for Rett syndrome using this antiretroviral therapy, which should be like an easy treatment for these kids. But moving forward, everybody asks, so if you can do that for Rett syndrome, what about these late onset age-related conditions such as Alzheimer's and dementia? That's a big burden on our society. And we are moving into that direction now, and we have a project with Alzheimer's where we are teaming up with my colleagues in Brazil at the University of Manaus. These are in the middle of the Amazon, where we decided to explore a little bit of the original tribes, the knowledge that they carry to create a new project. This was after an experience that I have with one of these tribes where I was interacting with them and asking them about what kind of a medicine they use. And they were telling me that the forests actually have such a biodiversity that they can cure everything as long as you know exactly what to look for. And so we start interacting, and I'll show you like the stages where we are now with this project. As I mentioned, these are really in the middle of the Amazon with the original tribes. They don't have too much contact with the white people, but we can go there and we can ask those questions. And they're pointing us to different plants. These are basically the knowledge from the old guys, the shamans. So this is one plant. I like that one because it looks like an eye, but it's guaraná. And we know one of the neuroactive compounds on there that makes you awake. But there is other hundreds of neuroactive compounds that was never studied by science. And the way they prepare this brew by mixing the different things was never been studied before. So we actually record the EEG from the shamans while they're preparing all of that. And to find where are these plants, we create like this swarm of robots that carry the knowledge of these ancient shamans that scan the forest looking for the plants where they contain the neuroactive and tells the tribe where the plants are so can they look for it. At the university, they purify these compounds, and we tested in the organoids here in San Diego to see if they are actual neuroactives or not. And the whole idea is after they pass all these quality controls to send them at the space station so they can be tested in brain organoids derived from Alzheimer's people. The way we're planning to do that is a different way. We are creating new forms of analyzing these organoids that was never done before. So this is a new way to, a new type of a payload where we have a microfluidic chamber in the center of these cubes. And each one of these cubes contain microelectrode arrays that can press down a little bit on the organoids and record the electrical activity. People always say it's impossible to do electrophysiology in space, but now we have a solution to do it, so we can actually measure and visualize the electrical activity of these cubes using this type of strategy. So we hope with that, the astronaut will be able to inject in each one of these cubes a different type of neuroactive, and we can see if they are protective or if it can rescue some of the senescence molecular pathway that we see activated in space. So there is more to that that we're exploring. For example, the applications will go into rejuvenation, regeneration, health, aging, and also drug screening. So we hope that we can screen drugs in space as well. So, so these are the main message that I have. Growing human brains in space can actually teach us about the impact of space environment into human cognition. We can take advantage of such accelerated aging to actually, in a compressed time, understand human diseases and to test novel compounds that were never screened before here on Earth. And I would like to plant a seed, which is just an idea into your minds, that the future is when the scientists will actually prepare these experiments by themselves. Right now, if we are relying on a commercial astronaut or in a tourist to do these experiments, we're going to fail. We need actually to train our own scientists to go to space and perform these experiments. And I'll finish here with like a futuristic view of one of the UCSD students, perhaps team up with a cost student to perform these experiments in space. So I'll acknowledge my funding agencies and members of my lab that are here who participate on this project. Thank you.
Okay, thank you, Alysson. Yeah, Alysson's always a really tough act to follow. So all I can say is I'm going to talk about dirt. That's how I feel after I follow you, Alysson. So I'm going to talk about a drug that is now under review by the FDA, where the development of this drug was accelerated by space, but it was originally discovered in dirt. It's a lowly soil organism-derived metabolite. So meaning this bacterium in the soil just kicks out this drug. So I'll talk a little bit about that, but mostly about space-based research, predicting and preventing accelerated aging and cancer stem cell evolution. Wanted to start by saying what a joy it is to have Michelle lead us as the executive director of our Sanford Stem Cell Institute, and to have T. Denny Sanford supporting us, and every one of you in the room for being here, because we're talking about a new frontier. It's not just Star Trek. We're actually doing it with our partners. We're going to democratize data, right, Chris? Dr. Longhurst is here to make sure you get your data that we learn about in space, as Alysson was alluding to, but really to say it takes a team. People say it takes a village. You see Rebecca Boers up there, Peggy Whitson up there, Bob Klein, the architect for CERN. You know, we have our whole astrobiotechnology hub from last year, the picture there, and then our chancellor, vice chancellor of health sciences. So it's about people, really. We've got an institute. We've got 6 centers, a brain tumor and neurorestoration program, which will become a center, right, Alex? Right, Frank? Yes. So we're working together to really accelerate discoveries and really look at what kind of track record have we had in space. We've had a lot of firsts in space. You know, Alysson was the first to do brain organoid experiments in space. Arun Sharma followed very quickly, but he'd already started to work on cardiomyocytes in space with Joseph Wu a few years before, actually, and learned about regenerative potential in space, and we heard about that earlier. We were the first to have a global astrobiotechnology hub here. Why is that? Because we think differently. Maybe the weather's good. Maybe we're a little more casual, but we don't take ourselves quite so seriously. So we have this integrated space stem cell orbital research center. We have been very fortunate to have funding from NASA, ISS National Labs. We've been able to develop new technology platforms, as you heard from Alysson, but this is really a collective opportunity to do an even bigger, better job with our partners here in the space community. Some of you represent NASA, others Space Force. How many people here are from a space company as opposed to NASA or ISS National Labs? Space company. Okay, we've got good representation here. No pressure. We've got Tatiana Kiseleva, and we have Ludmila Alexandrov. Somebody asked earlier, what about the media? The media is all over this. They may not be in this room because we like to keep it to our cozy friends here. But here's Ludmila Alexandrov on CNN about discoveries that are being made here. Why do we keep talking about regenerative medicine? As we get older, or if we get cancer, normal tissues degenerate. We have to learn how to regenerate them. I was on a flight coming back from Frankfurt, and there was a pretty bad medical emergency on the flight, and I had to say, we have to divert this flight. Not happily, But we did. And, the pilot said, can you sit in the first seat, which is there? And, so we have to land these regenerative medicine discoveries together. But it can't be a one-off. It can't be an emergency every time we try and get a new therapy to clinic. It has to be streamlined, and we have to work together to do that. And I did make it back in time for our anniversary because people were very nice and say, yes, you can get on the next flight. So we were talking about the market. This is for Anna Brady Estevez for Space Force. There you are. For people that see this as a developing market and an opportunity, I'm going to be super crass. There's money to be had. There's money in them there hills here in San Diego. So right now, the regenerative medicine market is valued about $32 billion. It's estimated to reach $500 billion by 2033. Why is that? None of us want to age. We want to stop this process. Please reverse it now. But stop the aging-related problems that happen in our heart. So Arun, you have to work on this. Sarcopenia, Larry, please get busy with these small molecules that are going to allow us to regenerate our muscles. We've got Tatiana finding out the good side of aging, that some people live to over 100. Why do they do that? We've got to find out about neurorestoration, the bioactive compounds, yes, please, and ways to restore the blood, which circulates everywhere, and prevent cancer, the number one cause of death for people under 85. So there's an opportunity here. We're going to let Jana talk a little bit more about this in the Q&A, and the panel discussion, biomanufacturing, commercial, intellectual property. What gives something value? What makes the patent examiner say, well, that's cool, I've never seen that before, that's not obvious? It has never been done before. Do it in space, it has never been done before. That creates new composition of matter IP, new method of use IP, new technology platform IP. Intellectual property matters for getting something funded. It's great to have a nice idea, but if everybody else has had that idea, the patent examiner is going to go, nope. Somebody else thought of this. Davide asks this every time we submit a grant. Is there going to be IP? We can make novel drug formulations in space. Space is actually better because of the homogeneity of how crystals develop. So the protein crystal structure of PD-1 bound to its target Keytruda led to a different way to administer that drug. It's a $12 billion drug annually. You inject it now instead of being tethered to an IV pole. Well, if you have cancer, you don't want to spend your time tethered to an IV pole. You want to get an injectable. That changed everything, and that happened in space. You can make some compounds that are hard to make on the ground because they have a complex structure, and that happens more readily in space. So there are opportunities here. And, this is the last time we met here. We had Sandra here. Thank you for being here again. We had Twyman here. We'll put you on the spot with lots of questions, Twyman. We had Jana here. Mike Roberts is going to be at South by Southwest. We had Rhea here, but we figured for this panel, natural intelligence trumps artificial intelligence. So here's why space, the astrobiotechnology hub. So the economic impact of life sciences in San Diego, you'll see it's quite large, 56 billion. If you look at the economic impact in greater LA, the whole Southern California area, we're around 66 billion. So that's over 100 billion in economic impact that we have by working on life sciences. Add space, and that's a whole new dimension. And it's all happening here on the West Coast. Thank you, our Space Force friends for reminding us of that. SpaceX for doing this, Redwire for being here. We've got vast space and new opportunities to work together on stem cell aging and pre-cancer. So as a cancer doctor, I want to know, everybody I see wants to know, will my disease progress? Or if it's supposedly in remission, when will it come back, and how do we predict, prevent, and reverse that? That's it. It's a super simple question. And a lot of this has to do with what Alysson mentioned, stress. How much does stress play a role in cancer initiation and progression? And that's what space has taught us. It has taught us when we're all stressed, we activate a very specific enzyme that's human-specific, and that enzyme is called ApoBec, and it's there to repress those repetitive elements or human endogenous retroviruses. And so we've discovered that those are activated when we're all stressed. We discovered this in the astronauts. Day -2 before launch, some of them activated these repetitive elements, the human endogenous retroviruses, and then they activated this enzyme, ApoBec, to block that. What's the problem with that? APOBEC introduces mutations into our DNA. So if it's on for too long, if we're stressed for too long, if it's too acute a stress or too long a stress, we make mutations in our DNA. That's pre-cancer. So that's an important finding. And when that's activated for too long, it makes what Alysson talked about, IL-6, interleukin-6, and that activates this other enzyme, ADAR. That enzyme allows cancer to clone itself. So that seemed very Debbie Downer-ish. And I don't want to know this until we started to look at space, stress, and longevity. And we talked about the NASA twin study. Scott Kelly came back 2 inches taller than his brother, so I wanted to go up there as soon as I saw that. But wait a minute, he had immune dysfunction, pre-malignant changes, though these mutations in his blood, and increased inflammatory growth factor expression. So we thought these are all the things that allow cancer to be initiated and lead from just a pretty benign precancerous condition to fully malignant condition. So we started to realize we could make technologies, this is together with Space Tango, Twyman, thank you for being here, something called canaries. So my dad was a coal mining engineer. And does anybody know why coal mining engineers used to take canaries into a coal mine? Anna?
They would smell gas.
They smell carbon monoxide. So when we can't detect it, they're gone. So it's not a very nice experiment. The canaries don't survive this. But we made these detectors to tell when our stem cells are stressed. So I could do this with all of us. Put your stem cells, this is thanks to Jessica Pham and our whole biorepository. We've got Inga and Jane and Carla represented here, Patrick. We can put our stem cells in these nanobioreactors and say, are your stem cells stressed? Are they dividing too quickly? And can we slow that down? So we have different reporters. One of them we made with Roger Chen, the Nobel laureate for GFP. So we can see in space, these are experiments with Space Tango, that stem cells are woken up way too much in space. So instead of all being red, sleepy stem cells, they wake up too much and they're exhausted when they get back. They shorten their telomeres. So we found that with Lyudmila Alexandrov. So those are the genes that keep our stem cells intact. And they make mutations based on activation of APOBEC, that stress enzyme. It just happens faster in space. Does it happen to us on the ground here? Yes. Did we know about this? Yes, we did, but it took us longer to see it in individuals. All the time frames are compressed in space. It happens faster in space. We get 5 times more mutations in space than on the ground, and we find that we unleash the dark genome. That my mom calls us the Darth Vader of the genome, because we've inherited that dark matter of the genome, the repetitive elements. It gets activated in space, and it activates APOBEC. So then we said, well, that's in this device. What happens in people? So this is together with Jana and Pinar and our astronauts, and including Rihanna Barnawi. So thank you, Ahmed, for leading her through her Ph.D. And we studied them. We looked at their single-cell sequencing. I think, Chris, you can help us put this in all of our data sets. We can upload some of these data to our portals as we start to show that they're rigorous, reliable, reproducible. And what we found by single-cell sequencing and other techniques before, during, and after launch is that their stem cells aged too quickly and they activated the dark genome. They became mutated. I'm going to get through this a little bit more quickly, but they activated that APOBEC enzyme that mutates DNA. And here, this is going to be hard for some of you to see, but it matters how long in space. So after 10 days in space, they had a few mutations. After 21 days, everyone had a lot of mutations. But I want to go back to Imed's point about resilience. A lot of these people get better. These mutations go away. Why did they go away? Because the immune system recovers on Earth, and it gets rid of them. The immune system can see the bad guys, and we can help the immune system to see the bad guys by getting rid of this immune silencing gene expression, ADAR. We know that that gene is responsible for metastasis, the tumor spread to other sites. You can see here with that green fluorescent protein. You can see it in these samples that Alex Khalessi has provided for us and is helping to lead the charge on treating people with metastatic disease to the brain, whether it's breast cancer, lung cancer, or a primary brain tumor. And we can detect that the stem cells are fed, the cancer stem cells are fed by normal stem cells. So this is one of the samples that Dr. Khalessi gave us here. It looks like an alien. It keeps growing. It's activated that cloning gene and cancer immunization gene called ADAR1. This is Teresa Esposito doing this in the lab. Alex Khalessi really showing us how to work on something that's really tough. And so we'll ask a lot of questions of you. But this is something we know how to study now, so what do we do about it? We found out we were fishing in the wrong place about how to block this pathway, and Jessica found in the lab said, we've got a drug that works. I said, Jessica, no, you're looking at the wrong thing. Well, of course she was right. Inga was right. It works in not just adult AML, myelofibrosis, but also pediatric AML. And Mike Burkhardt here, who's the head of chemistry and biochemistry, had discovered this drug through chemoenzymatic catalysis. We've been able to scale production. Our son Harris is at the back of the room. He thinks chemistry's cool. He thinks this is the stuff you do for fun. It's not that fun all the time, I gotta say. But it's fun when you do it in space. So this is Jessica doing our first experiment in space on the International Space Station, first to test a cancer drug in space. We've got that ADAR reporter in these little mini nanobioreactors. As Pinar alluded to, we saw cancer tripled in size in space, so it gives us a faster view of what cancer does under conditions of stress. This happens on the ground. It happens when people have their cancer progress. We just see it faster, and it activates ADAR. So this is one of the last slides. This is what it's like to have your experiment run on the International Space Station. Here's Walter Villaday showing the breast cancer organoids here. He's making sure they're in the right orientation. The day before, he was a little disoriented. So he didn't get that much out of that day, which is why it's so important to train astronauts, scientists here on the ground to be astronauts, and astronauts to know what our science is. So this ended up leading to the discovery of the fact that we've got a cancer kill switch. So we have a drug, fedratinib, that can reduce ADAR activation. It's now FDA approved. We worked on that in my lab with a local company, and we had to have a major champion for that. Thank you, Sandra. So there are people in this room who are repeat heroes, and they're all on the stage. So anyway, that discovery of rebexinib, named after Rebecca Morse, thank you, is possible because somebody believed that science matters. It wasn't one person, it was a very strong group of people, but it matters to be able to predict, prevent, reverse pre-cancer transition to cancer. So with that, I'd like to thank all of the NASA and Axiom crews, Space Tango, Sierra Space, Axiom Space, all of our partners, our funding organization, the lab. Thank you for not giving up on these ideas, Davide. Thank you. And then this is, Noah was here. Thank you for being here again. Alysson, always have great comments and insights. And then Sandra and Peggy. And then this is Alex. He's trying to save the life of somebody who's 22 years old who's in this picture. It shouldn't be that hard. It can't be a one-off, like helping to make sure somebody with a medical emergency gets the attention they need. This has to be a collective force. Space Force, ground force, whatever it takes, we've got to do it here. This is the first launch of tumor organoids into space, 2, right there. 1. So some of you have seen this video. This is the first time tumor organoids had ever been launched into space. And Alex Khalessi and Chris Longhurst and I just watched a Starship launch with Paul and Elena, and it was even better than that. There was a double sonic boom, I think, and it was caught. But really, this is going to take investors, philanthropy, and academic convergence. It's all working together because we want to be here. All of us want to be here. So it's for you, Sandra. Thank you. All right. So with that, I really want to make it interactive so people in the audience can say, hey, what about this? I have a question, and you have to answer it right now. So we will. But I wanted to really start by asking Alysson, because he's made such tremendous technological discoveries and forced us to all work on this. You had quite a futuristic vision there. What do you think is the toughest challenge in terms of translating the discoveries that you've made in space to relevance to us on the ground here? What do you think that high uphill battle thing is?
Yeah, I mean, there's different things, right? I think the first thing is to convince ourselves that what you see is real. And so I echo the previous panel that suggests that you have to repeat the experiments. That's why we do multiple payloads. But that's not so easy from the logistic perspective, as well as on the financial side. I mean, it's very expensive to do it. But once you have that, and you are ready to translate the science, I think that's the fun part. That's what we do best here, right? That's why we have like the Alpha Clinics, Sheldon, and all the team here that can help you to translate. So I think we are in a better position because we have the whole team to facilitate that steps. So yeah, two things. I mean, first, convince ourselves that the space data is real and can be translated. All the preclinical data derived from the space should be done on Earth, and then move into clinical trials. And I think that's our advantage here.
And you've been quite a leader in terms of getting global partners. I mean, you've been in Brazil, you've been in Saudi Arabia. How do you develop those international synergies?
Well, it's natural, right? I mean, I get invited to talk in different places, and then we are dealing with problems that affect the entire world, right? I mean, aging is a major problem. I was just in the Emirates talking about aging as well, and it was super fast. I mean, I give like a talk in one day, and the next day there's a bunch of people who wants to team up and see what we are doing here, understand better what we are doing. So I think there is a conversion, because the problems are for all the nations. I mean, Alzheimer's is a big deal everywhere. Aging, dementia, nobody has a cure. Nobody has a model to study these conditions. We are providing a model system. And I think that's very attractive.
So speaking of model systems, I'm going to switch to Dr. Khalessi. How important are these humanized model systems for understanding that deep biology that only you have access to as a neurosurgeon?
Yeah, well, I mean, first of all, thank you. And that's a big part of the reason why I'm so excited to practice in this ecosystem, because I do feel a very special responsibility to learn as much as possible from every time we access the living human brain. And what I thought was so beautiful about your presentation, Alysson, was the fact that if you look at the extremes of life, most of the conditions that we don't have the opportunity yet to deal with as neurosurgeons are those neurodevelopmental and neurodegenerative conditions. And so when I look at a research question, like the questions that this group are asking, it's, what puts us in a better position to answer that question than we could before. And if you relax the constraint of time scale, you're able to really iterate that cycle time much more quickly. And that's what's so attractive, because you're learning much faster what works and what doesn't work. The other thing that was embedded in your presentation, looking at those different neuroactive compounds, is we now have computational power and the ability to scale by trial and error in a way that's really unique and different. But once you actually identify those neuroactive compounds, there's a whole environment, an ecosystem here that's really looking at delivery of how an integrated system works. And so as outstanding and amazing and responsive as your organoids are, they're quite a different thing than the unique phenomenology of the human brain. And so once we actually identify that kind of funnel or that superset of compounds, it's going to be important for us to then identify the network effects in the brain, and when we intervene, with what compounds in what way, with the right time scale. And I think that that's an area where neurosurgeons are going to have an opportunity to really contribute. I'd lastly say, just because he showed one of my patients and some of the work that we've done together around metastatic disease, one of the reasons brain metastases are such an attractive model for the power of this kind of work is it used to be 10 years ago as a neurosurgeon, it was rare that you actually operated on cranial metastases, because that was largely considered a terminal event. But now our systemic treatments of cancer have improved to the point that there's a lot of value in taking out that isolated metastasis. So it's a unique opportunity to understand what I consider kind of, in an adverse way, biological super warriors, right? Because if you imagine what a metastasis has to do, it has to actually travel and hazard the bloodstream and the immune system, implant in the central nervous system, and then outcompete tissue that was actually designed to be there. And so I think there's a lot of insights that we can actually gain from CNS metastases that don't just have the impact to help a lot of people, but also will allow us to learn fundamental lessons about biology that will scale to a host of different diseases.
So why space?
Well, I think partly for the time scale issue that we talked about. And also, I think in the earlier panel, or when you actually think about this kind of real enthusiasm for payload science, that is to actually do experiments that can actually benefit folks on Earth, but also when you think about the impact on the physiology of astronauts, or when we think about space travel, a limiting step to a lot of that advancement is going to be the impact on the central nervous system and microgravity, and specifically venous side disease. And so I do think that that's ultimately going to be a limiting constraint as we take a more expansive view of space and human health. And so I think it's really important that we actually fairly early on start to learn as much as we can about those changes, because I think in the absence of that, that's ultimately going to be a limitation. There's so many, I think, great role models in this environment, and I've learned so much just by participating, because if you don't dream it, it definitely won't happen. And so I think the fact that we really challenge assumptions here is really what's made it a very exciting environment to practice.
So I'm going to, speaking about dreaming, switch to Rebecca Morris. Becky, what is it as a community leader and philanthropist, don't worry, that makes you want to help other people dream? Because our dreams wouldn't happen without support, We wouldn't be inspired to do things, but, what allowed you to get involved? I mean, you've got your name on the Rebecca and John Morris Cancer Center here. You've supported people, our purpose and programs. What makes you keep doing that?
Well, I want to say first, there's a big difference between me, and a unique difference between me and this panel, and that is, okay, that's it, my memory. I'm going to have to use some notes to help me. See, I almost forgot what I can't remember. So I don't know if I'm going to answer exactly what you said. But we all know here that cancer is one of the most prevalent and serious diseases that we have. And there's already a lot of research, I guess, going on, thank goodness. But we have really an outstanding, as you were quoting, I guess, all of California, an outstanding resource of medical institutions. And I came from a city, Houston, which I don't know or Texas has what we have, was my home for about half of my life, but I was familiar with I'm lucky to be in communities that did have really good medical resources. And I've known Kat for a long while and feel like she's one of those best resources that we have here. And if you know Kat, and I assume that most of you do here, she's an irresistible force.
Very tactfully put.
He most knows, perhaps. So when I was looking, when I was looking for someone to be a recipient of a gift or a contribution, I didn't have to look any forward to her and her lab. I know, I don't have to read now. I know she will continue to contribute in her lab, which is a big part of her work. I'm sure she would say also to medicine. So in this case, that's how I decided on a contribution. I can't say that there is any pattern. I don't know. Some people maybe have an area that they're most interested in. I know my former husband was certainly part of the original cancer center.
That's right.
And he always felt that medicine was the best recipient of a contribution, because he felt like that, and I'm not saying I didn't agree with him, that the results were long lasting.
Right.
And affect so many people. I happen to have the conflict for me was I had a tenderness isn't the right word, I'm not sure, in my heart for children, as many people I'm sure do here. And you can see a hungry child, right. Are you going to say, oh, I'm not going to contribute to them, right, and give them a sandwich? And so that has a little conflict. So my philosophy has been sort of to spread my interest. There are other things that do relate to children. I would say it would be secondary, or at least up there for me, but everybody's personal.
Yes. So, thank you so much, Becky, for being here. I know that you did. So medicine matters, as Becky so aptly put it. It has a ripple effect. It improves our community. It makes us all stronger. So I'm going to switch to Sandra about medicine. Why does it matter to make new medicine or advocate for new discoveries, whether it's diagnostics, therapeutics? How can space help us here, or does it? Is it just a distraction?
Sure.
I think that, for patients, especially those of us fighting, sort of long-term cancer. I've been battling with my disease for quite a while, and early on, I was confronted with very little in the way of treatment options, and those options were quite daunting. Very, very painful, almost as bad as the cancer itself. And I know that I'm not alone. For those that have friends and family that have struggled with cancer, that being faced with the decision of treatment and how difficult treatment itself is, that stuck with me very early on. And when I came to know Dr. Jamieson and was able to get on her drug trial, I was at first very, very afraid, thinking that I was going to be confronted with a lot of struggle and a lot of heartache. And what I found was it was like a walk in the park. I felt like I kind of cheated, and at some point, I was like, is cancer gonna get me? Like, what is happening? Why is this so easy? And that feeling of this is what treatment should look like, and this should be just baseline, for everyone. And even above that, why did I even have to suffer with cancer for as long as I did? Like, to nip it in the bud before it even took hold. So when you talk about this just amazing work that you're doing, and this idea of timeline, condensing timeline, I think about iteration and how being able to turn that research and grow that understanding, quicker and quicker. I think of the voice of the patient inside of that, because you might come to an answer that's very hard on the patient, but you can go back to the drawing board and come up with an answer that's very easy on the patient. And that's something that was unheard of before. And now you have that opportunity to both defeat cancer and make it really easy. Like, make it a spa day. Come on.
A spa day. We do have massaging clinic, by the way. So speaking of the space-time continuum, I'm going to turn to Jana, because when we first started doing these experiments in space and we were witnessing the astonishing success of Alysson's experiments, plural, Jana said, watch out, it'll just be different. I just didn't anticipate that it would be bigger, better, faster, smarter, that things happen more quickly and you get new answers. I don't know if it's a spa day yet, but we're hoping it's going to be a spa day. What are your thoughts, Jana, about the global astrobiotechnology economy, how we do this on steroids to get everybody here working together to make this happen faster?
Well, first, thank you, and it's so nice to be part of this panel. I'm just touched by everyone that's here and the commitments that everyone has made. And I learned last night that Rebecca actually had an internship at NASA, so she's not that far away from our space community. So
I didn't want to put Becky on the spot.
I was I got to U of H for 1 year. I lived in Houston, went to the University of Houston. Somehow heard that there was an internship. It was 60 years ago. I don't remember how, and it was competitive, but I don't remember how. So anyway, I got the internship. You supposedly had a partner and you alternated. You went to school one semester, they worked, and vice versa, so you could continue your education. And unfortunately, oh no, I shouldn't say unfortunately, but fortunately I got married.
Freudian slip.
And did continue school as my husband did, but but I no longer could do the internship, and I often wonder if things had been different, if I would ended up with a job there, not as an astronaut or an engineer. I can hardly tell you that. I'd had to learn typing or something. I don't know what I would have had to do, but it was an exciting time for me just to work in that environment.
And here you
Yeah.
are back in that environment.
know.
So space never leaves you, right, Jana?
Right,
Yeah,
exactly.
thank you.
Well, and I think for me, one of the things that I really resonate with is medicine informing novel discoveries. And when you think about space and how space can inform novel discoveries, when we find out how the body responds the way that it does in that environment, or how cells respond, it really can give us new insights and help to accelerate. And to the point of new IP, and when you mentioned, opportunities, we like to think of the Integrated Space Stem Cell Orbital Research Center as kind of the next nexus of biotechnology 250 miles up, because that's what's going to happen. We're going to spin out more and more companies and technologies. It's what we have here on the ground, but we're just taking that a little bit off planet. And I often say that I really, in my world, the definition of global includes 250 miles up. And that's what we're working on now. And that's how we will accelerate and help to really make it a spa day, I hope.
So with that, I just wanted to see, are there any questions from the audience? Because we want to get to the reception so that everybody can mingle and learn from each other. Any questions out there? Because this is an unusual constellation of stars up here. Anything? Yeah, Christina.
Thank you. I always come here and my ideas are also untethered, and so it really opens up my mind. I'm just wondering, in this studies of the organoids and some of the next steps to look at inflammation, are any plans to put in co-cultures of organoids with lymphocytes, or have lymphocyte inflammatory behaviors being studied already?
Yeah, so we did that on our last launch with Axiom Space. We added the stem cells that produce the inflammatory growth factors with the cancer cells. So Pinar and Jana were actually both part of that, and it did promote cancer growth. But Alysson and I are working on the role of inflammation from our blood on the brain organoids. So maybe you want to comment on that.
Yeah. I think both the immune system, the peripheral immune system, as well as the immune system from the brain, the microglia cells. So our last payload already incorporate those cells. We call neuroimmune assembloid. So it's no longer like an organoid, but an assembloid, because it combines the different systems in the body. Yeah, so we continue to improve the model, because the more close to the reality, the better the answers we have.
And speaking of closer to reality, so those experiments that are happening here are going to inform who would most likely benefit from a Parkinson's disease trial which has dopaminergic neurons. Will inflammation reduce the chance of having a good response to that? And that's technology that was developed by Shinya Yamanaka, who got the Nobel Prize for inventing induced pluripotent stem cells. But we have to know, when are we wasting people's time and it's not a good spa day? When is it a great spa day and, Parkinson's disease is not a problem? It's in the rear view mirror. So that's what Alysson's technology is helping us to understand. That's why understanding stem cell aging in centenarians, why don't they get these things? This is what Tatiana is doing. David and Tatiana are working on the liver, because we've talked a lot about the blood and the brain, but what about the liver? What about muscle? What about being able to make better stem cells? Can we train them? Can we, maybe they're reformable. Maybe they don't always act badly. Maybe we can get them to take a break and not be misbehaving all the time. Any other questions? Anyone? Oh, yes, in the back.
Hi, I was wondering what is the key characteristic of the space environment that is generating this accelerated aging? Is it the fact that the organoids are in free fall, or is it the radiation from the cosmic rays? And then in both of these cases, could we mimic those conditions in Earth?
Yeah, I'll take that first, and then Alysson has quite a long answer. Yeah, so we've seen you can't mimic this on Earth. Microgravity is unique. There's radiation up there that isn't just ionizing radiation, and Sylvain can comment on this more. There's also other particles, protons, neutrons, other exposures that are happening up there on the International Space Station. But also the microgravity allows for three-dimensional assembly, very much like in the developing embryo, as Alysson was talking about. But that is what happens in a developing tumor. The tumors think they're embryos. They have a three-dimensional structure, and they're allowed to do in space what they do in people in a faster time frame. So none of the microgravity simulation experiments showed that. We only learned it in the environment that Jana told us we had to work in, and Alysson said we absolutely will learn things, and NASA said we have to do it, and ISS National Labs said we'll give you the space on the ISS, and now we have companies that are going to give us a lot of payloads. Thank you, Paul, for doing it. And then for automating it, Twyman. We still need astronauts, but we have ways to make new therapies with red wire. So I think it's just an important environment. Alysson, what are your thoughts?
Yeah, I think it's complex. We cannot isolate a single variable. In the beginning, we thought that it was all about microgravity. I don't think that anymore. I mean, there is microgravity, there is cosmic radiation. You cannot reproduce here on Earth as well. There is magnetic fields that changes. There's so many variables that we cannot control yet. That's why we are broadly calling space environment, because it incorporates everything. With time, we'll be able to isolate each one of these variables to figure out exactly which one is causing some of these effects. And it might be a combination.
I do want to say one thing. It's teaching us a lot about stress-induced aging, cancer development that happens before the astronauts leave the ground. So the question is, how much of our cancer initiation and progression are part of a fight or flight response where we think we're going to be under tremendous stress, and how do we reduce that, Sandra, so it's a spa day? How do we de-risk this so people know we have better therapies? I'm looking at Davide, because we're going to rely heavily on you, so that they know there's a back pocket solution, there's a front pocket solution, and we're going to be able to apply them to everyone, and this therapy is going to be accessible, and we know the right person at the right time to use these therapies in, and that's where Chris Longhurst comes in, no pressure at all. But this is what we're going to be doing together, if we have our way, collectively. Yes, our newest faculty member to the Division of Regenerative Medicine, her first day. Hello. No pressure.
Thank you.
Hello, Elsie.
So thank you so much first for creating this full circle. Science plus people that donate their money and patients and physicians. It's incredible to see the results of a communality efforts. This is like emergent properties that came from a team of work. So thank you for doing this. And I have two questions. The first one is about if you have think about studying developmental biology in space and using synthetic model organisms, of course. And then the second one is about the circadian rhythms, and then how important they are for us, for our physiology, and how these that we don't have in space, I think, impact on the physiology of the organoids. Thank you.
Yeah, so the circadian rhythms is quite important. We think that the organoids intrinsically have a molecular clock. So they kind of figure out when it's night and day. And you're right, at the space station, this is all messed up. So we are beginning to understand here on Earth before we try to explore that in space. Tell me again, what's your
reproductive developmental biology?
Yes,
that was the question that Ahmed had earlier.
Yeah.
Yes.
And by the way, I do have your answer, because in our first mission, we actually sent organoids that are quite young, and they were just developing, and the brain actually develops in different ways. So having a baby in space is not good. So don't do it.
That's a problem. That's a problem for colonization.
It is a big problem. Yeah, if we don't solve that, yeah, there's going to be... And you know what? I think it's so funny because I read lots of science fiction books and nobody talks about that. This is something that the science is bringing to the community. We cannot reproduce in space. Or if we reproduce, it's going to be like a different species. Maybe we have an octopus-like brain instead
Maybe
of...
other countries did it already.
Oh, we don't want to know. Well, that would be an interesting way to end this session. It's global. We're going to go back to Space Force. Hello there.
I got one more. So
Yeah.
2 years ago, I was at the University of Colorado Boulder, and they briefed their graduate school engineering does a lot of stuff with Space Force, and a lot of the launch providers and rocket companies as well, looking at how space adaptability for the human brain and human body in space as you go past this lunar. One of the experiments they did, and I can get that for you, I can talk to the professor and get that for you, but one of the experiments they looked at is when you get past this lunar, the electromagnetic waves between the moon and the earth, that gravitational pull has a direct, it kind of tricks the brain a little bit about how it functions. And they're looking at ways to get it to some kind of, similar to the circadian rhythm conversation you just made. And they're trying to figure out ways to get the brain to say, hey, trick it back. You're okay. You're in orbit. Everything's okay. You're making the moon unsafe. There's nothing happening with you right now. So I think that has a play with astronauts too, as they travel between the different LEO, GEO, MEO, and they move out of orbit, or they're doing maneuvering and things like that too. So I didn't know you guys had looked at that perspective of some of the research you're doing. Thank you.
Does it work on the ground? Yeah, so cislunar near the moon, that's what that means. So how are people going to behave in terms of their brain function, whether they're close to the moon or on Mars? It's going to be different. And developmental biology, not just for when we start from this little thing, developmental biology happens continuously throughout life. We're always regenerating our tissues in response to injury. And that may be impaired in space. It may be better in space. We just need to learn. And what environment is the best, we don't know yet. But it's a matter of gravitas. We will find out. Any other questions? Anna.
Anna Brady Estevez. We're establishing a new deep tech fund, American Deep Tech. And so my question would be, there's such, just really want to applaud this extraordinary work, so much inspirational work. How do we as a community bring forth the resources that are needed financially, human capital, whether it's digital AI? What do you need, and how can we, as a community, extend that voice, be it to the workforce, talent, investors? What would you share?
That's a perfect question, Anna, to end the panel. Thanks for bringing that up. So I'm going to let everyone answer that question. So beginning with Becky, how do we invigorate other people to start getting involved in science and supporting it? Is there a way you get people to say, hey, science matters or medicine matters? You're one of the most convincing people I've met.
Well, I really don't know anything other than communicating with people, perhaps talking to people, and try to get them to think about it.
Yeah.
But
that's the key.
That's all I would be able to say.
Well, that's the key. It's communication. Sandra, how about you?
So I tend to think of science and research and this brilliant type of collaboration where folks gravitate towards problem solving and towards creating something new. It's kind of the best of us, right? The best of who we are as a species. And so, gathering people towards that greater good, and the need that I think many of us have to be a part of something, to contribute, to be a part of something bigger. This is an avenue that I don't know how you get bigger than this, right? And the opportunity there is so incredible, and so that it's a, I just get very excited thinking about that. just this chance to make a difference. And, looking back on, the shoulders that we have all stood upon to get to where we are those shoulders, to the future generations. Heed the call, right? Science is this means, space is this means. I'm very optimistic, but I also need that confronted in crazy days where sometimes we all feel helpless, right? We still have each other. We still have this. Yeah.
Yes, Becky. Yeah.
Is it Sandra? She may have said this, but it just occurred to me that those people, I do have a medical issue, which is how I met Kat. And you do, I guess that's what I understand, that people maybe that do should share their experiences more about whether it's helping themselves by listening to your body and getting early intervention. That's really important. I was very fortunate in my disease. You said that I don't know if it was early, but it was a low-level disease.
Exactly.
So, and I had breast cancer, same thing, was found very early, had a good prognosis. And so sometimes I know when I first realized I had it, I didn't want anybody to know, because I didn't want to be seen as a person with an illness or with cancer. And so I wanted to keep it secret, even from my family. And then after a while, I decided that was sort of silly. But there may be other people that feel that way, that don't share problems that they have. So I think both people that have had difficult times and can share and suggest getting regular medical intervention, and people that have had easier times, which I feel I have, can share that there's that possibility too. But again, really important to get to your doctor. And
so remove the fear by communicating, and let your family in on it. We need to share, and we need your support, Anna. You could see this. It's deep tech, but deep thinking, deeply felt convictions that we have to be the generation that helps people. So thank you, Becky. That distills it down perfectly. Heed the call, as Sandra said. Don't be afraid, because we have new things. If we don't have them now, we'll make them. Becky's got a drug named after her now, Rubexinib. So we will.
Not everyone.
Not everybody can have a drug named after them, so it's a little bit of a high bar, but she makes it look easy. Alex, what are your thoughts?
Yeah, so I appreciate the question, and I actually think there are kind of two very distinct value propositions to this type of work. One is fundamental and descriptive, and progress in science is going to be nonlinear, and that's going to require a distinct ecosystem that actually understands that the result of that work isn't going to have an immediate deliverable or even a deliverable on a short time horizon. So that's going to require public-private partnerships and a risk tolerance to actually take on those things, and making sure that the work's done at the right level of analysis. So the question about circadian rhythm, obviously a lot of the organism level work that we're doing now is on the astronauts themselves, and actually making sure that we're learning as much as possible from each human that goes into space. And that's foundational work that's going to require a lot. Then the second piece is going to be the development of targeted therapeutics for specific diseases. And so that's going to require organization around those specific diseases, be it cancer or metastases, neurodegenerative or neurodevelopmental conditions. And I think those teams of people are actually going to need to answer those hard questions of what precisely needs to be done in space and what questions need to be answered here in a coordinated way. And it's going to be those kind of distinct research environments that are going to develop. Because I often think that in neurosurgery, this has been a kind of a common refrain, that even when you find a technology that works or a treatment that works, is making sure that you identify the subset of patients in which you can actually demonstrate that benefit is often the larger challenge. And so I think that in the work that Alysson showed, it really beautifully demonstrates how we can kind of screen those candidate compounds. But then the important work that's going to remain, for example, like in your Parkinson's example, is which Parkinson's patients would potentially actually benefit from this space-generated therapy. And so that's the way that I actually see the broader environment developing: fundamental science and descriptive science, and then disease-specific for lack of a better term, tiger teams that involve space and earthbound, ecosystems.
Great. And then Jana, what are your thoughts? I know you have many.
Well, and I think that, what you said is so important, because even when we think about space, there's the nation science, so the fundamental science that needs to be done, and then there's science to build the robust commercial space economy, which is the translational piece that we talked about. And I think both are important, but to the question of deep tech, space is another tool in the deep tech arsenal. And some of those same components that we need to do translational work in that environment, we can also do fundamental work with. So what I would say is that as we look to the future, build those capabilities that allow us to do both types of science in this unique environment, so we really can accelerate and move the needle. And the public-private partnerships, the philanthropic contributions, that's what it's going to take. I mean, when I think about even, you made the comment earlier about, well, all the questions are answered, right, in science, which I just loved. And it's like the same thing in the pharma industry. All of the easy things are solved. The hard things, everyone needs to come together to do, because it takes that entire village, and it takes everyone's support and funding from all levels to be able to achieve that.
Thank you. And finally, Alysson, yes, thank you. We're going to let Alysson have the last word, because deep tech.
Yeah, I think we should be very persistent on social media. I think we should be very aggressive.
You mean TikTok? Which kind of
and then TikTok is as well, yeah. And I think our faculty, our scientists should be in all talk shows across the nation. We should be talking about science every time, all the time, and facilitate the communication. Learn how to make that more digestive so people can comprehend. Because once people get it, I mean, they love it, right? I mean, the problem is that
we do.
We talk science to each other and barely gets out. But when people get it, I think they appreciate the value of that. They understand where the drugs are coming from, where the medicine are being made. And I think this is all about how we communicate to the public. So we need more training. We need more people out there talking about science.